the Green Thinkers Academy.
Hello everyone, I am Nabanita Chakraborty and today I will be discussing about cell. And in this presentation I will be covering the cell structure, its function and the types of cell. Now what is cell? The basic unit of life, or it is called the building block of life. Living things are either unicellular or it can be multicellular. So from these two words we can know that unicellular means it is made up of a single cell, whereas the multicellular means it is more than one cell. Now the cells can be of different shapes and size. It can be oval, it can be elongated, it can be like long structures as present in the nerve cells. So here are some examples on the screen. You can see that the smooth muscle cells, the ovum, the sperm, the nerves, these are all single cells and have different structures. This cell comprises of a number of organelles which are present in it, and each organelle has their specific function. Cells also have the self-replication property in them, due to which they grow or increase in number. So the different organelles which are present in the cell are the ribosomes, the mitochondria, the nucleus, the plasma membrane, is the place where it grows, all the organelles are present. It also consists of the vacuoles, the chromatin, the Golgi apparatus, the endoplasmic reticulum, the cytoplasm, the lysosomes, a lot, and each have their own function which regulates the different functions of the cell. So basically we can say that cell is the body of a car, and the car which have the different parts, the brakes, the accelerator, the engine, the gear. So these organelles are all these components which together gives or drives the cell. So who discovered cell? So the cell is discovered by Robert Hooke, and it is done in 1665. So Robert Hooke, he has studied a cork and he has seen box-like structures in them, and he have used the old-fashioned microscope, and thus he concluded that these are non-living in nature due to you can say there is no development in the microscope. That is why he could not see that whether it is living or not. So next it came to Anton van Leeuwenhoek in 1674, who finally said that it is a living component. He has studied in pond the bacteria, and through which he has seen the movement in the cell, and therefore he concluded that the cell is a living component. Now the contribution of the other people in the field of the cell theory. So it is given by Schleiden, Schwann and Virchow. So these are also the three eminent scientists who have their contribution in the field of cell theory. So what did Schleiden say? He was the first one to note that the plants were made up of cells. Then came Schwann. He concluded that all the living things, they are made up of cells. Now the third, Virchow, he said that all cells comes from the cells. That is, basically he tried to explain the replication nature of the cell. Now the characteristics of the cell. So all the organelles, they are present in the cytoplasm of the cell. So cell is space in which cytoplasm is present, and on them the organelles are seated. Okay. Now the cell, it provides the structure and support to the organisms. Now I will be discussing the various organelles present on the cell and their function. So first is the nucleus. It holds the genetic information in it. Next is the endoplasmic reticulum. It holds the internal organization of the cell. Now third is the Golgi. It packages the proteins. and works in coordination with the endoplasmic reticulum. Next is ribosomes, which is the factory of the protein. So there is the place where the protein is synthesized. Vacuoles. Vacuoles are the places where water and the waste material is stored inside the cell. Next is the cytoplasm. So it is the space where all these organelles are seated. So what is the cytoplasm? It is the cytosol and the organelles together. We call it the cytoplasm. Our other organelles are present in the cytoplasm is a centrosome. So it regulates the equal distribution of the DNA. Next is the lysosomes, which is also called the suicidal bags. So these are basically the digestive enzymes which eats up the germs or the outside materials which are unwanted in the cell. And last is the mitochondria, which is the storehouse, the powerhouse of the cell that provides the energy to the cell to work. Now comes the types of cells. So there are basically two types of cells. One is prokaryotes and the other is the eukaryotes. So this one's karyot. This karyot word means nucleus, and pro, this means old, and eu means new. So prokaryote is old nucleus and eukaryote is new nucleus. So now we'll be discussing the features of a prokaryotic cell. So it is basically found in the old or the ancient microorganisms. They are found in bacteria, mycoplasma, pneumonia-like organisms. They do not have a well-defined nucleus, and we can call that, we can see a nucleus-like thing present in the cell. You can see the structure there. It is called a nucleus. It is not called a nucleus. Nucleus is a prominent structure which we will find in the eukaryotes, but in prokaryotes we can see the nucleus. Next is it has a naked genetic material. So it is not enclosed in something. It is open in the cytoplasm. It is smaller in size compared to the eukaryotes and multiply rapidly. Next is their hereditary material is a DNA or a RNA, and they reproduce by the process of binary fission. So therefore, they have no sexual way of reproduction. They are fully asexually reproducing organisms and reproduced by binary fission. So this is a simple structure which is very easy to draw, and while describing you need to write the points and to draw the diagram with proper lettering. Next is eukaryotes. So as I told you, eu means new. So these are the organisms which are not ancient and found in the recent history. So they are developed from the prokaryotes. So what are the characteristics of these eukaryotes? First is you can find them in protists, animals, fungi, plants. Here the nucleus is proper and present with a nuclear envelope. Next is they have an organized genetic material. So the genetic material is not scattered in the cytoplasm. It is present in an organized matter. The cell size, it varies from 10 to 100 micrometers. Now, the cell wall is present in some eukaryotes but might be absent in some. Reproduction, they do via sexually as well as asexually. When the plant cells, they have chloroplasts, central vacuoles, and plastids, but the animal cells do not. So both this plant and the animal cells, they basically fall under the category of the eukaryotes. Okay, and these vacuoles, chloroplasts, and the plastids are strictly restricted to the plants and do not belong to the animals. So now this diagram is there, through which you can see the various organelles of a cell, and here is a nutshell you can get all the functions of the different organelles of a cell. So the nuclear envelope, it is a membrane enclosing the nucleus and the protein lined pores present in them, allows the material to move in and out of the nucleus. So basically it helps in exchange of materials. Next is the chromatin. The DNA along with the associated proteins together is called the chromatin. Third is the nucleolus. It is the condensed region where the ribosomes are formed. So it is the birthplace of the ribosomes, and the ribosomes, what do? They are the factory of protein synthesis. Next is the peroxisome. It metabolizes the waste. Next comes the endoplasmic reticulum. So there are two types. One is the smooth one is the rough one. How do we differentiate the smooth and the rough basis on the presence of ribosomes? If ribosome is a present, then it is rough. So it is a specific, you can think like it's a surface, and if ribosomes are small dots. So on the surface, if small dots are present, and you can feel that it is rough. So it is a rough endoplasmic reticulum, and in which the ribosomes are not present, the cell is smooth, the wall is smooth, and it is smooth endoplasmic reticulum. The function of the smooth one is to make lipids. What is the function of the rough endoplasmic reticulum is associated with the protein synthesis since it contains ribosomes. Now, for this, this was a part which covers the nucleus, all the parts of the nucleus. Now it comes to the cytoskeleton of this cell structure. Under the cytoskeleton, we are covering the microtubules, the centrosome, the intermediate filaments, the microfilaments, plasma membrane, lysosomes, Golgi apparatus, cytoplasm, vacuole, and mitochondria. The mitochondria, it produces the energy, vacuoles, it stores the waste, Golgi apparatus modifies the protein, plasma membrane is the space, and is the membrane which allows the selective permeability of materials. It involves exchange of the materials takes place. Microfilaments are there. It is the fibrous protein which forms the cellular cortex. Next is the intermediate filament. It is also a fibrous protein that holds the organelles in space. Next is the centrosome. It is the microtubule organizing set. And last is the microtubules, which forms a mitotic spindle and maintains the shape of the cell. So this is the whole unit that is present inside the cell, and you can define all these things, the nucleus parts which I told, the cytoskeleton together, you can merge up to explain what is role and what are the functions of a cell. So now we will go on differentiating the prokaryotes and the eukaryotes. So first, these underlined words are those on the basis of which we will remember the things. So first is where it is found. Now, these prokaryotes, these are found in bacteria, mycoplasma, plurinuclear-like organisms, whereas the eukaryotes are found in plants, animals, and fungi. Next is no well-defined nucleus. So basis on the nucleus, prokaryotes is not well defined. So it is basically the nucleoid. Whereas in eukaryotes, we have a proper defined nucleus, that too covered in a nuclear envelope. Third is the genetic material. In case of the prokaryotes, it is a naked genetic material, whereas in case of eukaryotes, it is an organized genetic material with DNA and proteins with histones also. Okay. Now, next is a chromosome. Prokaryotes, they have only one chromosome, whereas in eukaryotes, they have more than one chromosome. Next is cell size. The size of the cells in case of prokaryotes vary from 0.1 to 0.5 micrometer, whereas in case of eukaryotes, it is 10 to 100 micrometer in size. Next is the process of reproduction. It is asexual in case of prokaryotes, whereas both asexual as well as sexual in case of the eukaryotes. Next is the cell wall. The cell wall of the prokaryotes are very rigid. Okay? Whereas in case of the eukaryotes, it is not so rigid, and it is not present in all the eukaryotes. That also you should keep in mind. Now the organelles. The membrane-bound organelles are absent. It is not there in case of the prokaryotes. Eukaryotes, they have them. Next is these prokaryotes, they are unicellular except in cyanobacteria. We should keep this in mind. We should note this thing that all the prokaryotes are unicellular except the cyanobacteria. It is one of the major important points. And whereas the eukaryotes, they are usually multicellular in nature. Next is the organelles, that is the Golgi, endoplasmic reticulum, plasmids, lysosomes, all are absent in case of prokaryotes, whereas in case of the eukaryotes, they are present. So, and you can say that old they were not developed, the new one are developed with presence of all these organelles. Then we have this ribosome, that is prokaryotes, they have a 70S ribosome and eukaryotes is 80S ribosome. So what is 70S and 80S thing? Basically the ribosome is made up of two units. Okay? You can see it like this kind of structure. One is like this and the other one is like this. So the mRNAs, so that is the place where proteins are synthesized. So it passes in between this, okay? They pass in between these two units and finally the proteins are synthesized. So this unit together we call it the 70S for prokaryotes and 80S for the eukaryotes. Now one unit is 50S and 30S. This together, though 50 plus 30 is 80, but this unit becomes 70S in case of prokaryotes, and for the eukaryotes one unit is 40S, other unit is 60S. Though 40 plus 60 is 100, but in case of eukaryotes it is 78, but in case of eukaryotes one unit is 40S, other is the 60S. Though 40 plus 60 is 100, but together in the case of eukaryotes it is 80S. So you need to remember that the prokaryotes have a 70S and the eukaryotes have a 80S. How is the 70S forming? With 50S plus 30S. 50 plus 30 equals to 70, not 80. And in case of eukaryotes, it is 80S with 40S plus 60S. So friends, I hope you understand the lecture, and in case you have any queries, please comment down.
The Green Thinkers Academy.